Today we're going to use this lecture time to basically review what we've learned so far to prepare the midterm exam 1. So this is going to be the basic summary of all of them for the first or third part of the course. And rather than I go through all the details and talk about all of the features and the structures and some more about properties that we covered so far, I want you to actually go back to the pre-recorded lectures if there's anything unclear or if there's anything you think you missed. And also, you can come to the office hour and talk to me and ask questions too. So I hope I can just cover pretty much everything that you need to remember and review during this session, especially for preparing the exam 1. And again, always, anytime, you can email me or come to the office hour using the Zoom meeting. So I just want to remind you that we had introduced in this course description as well as goals for this biochemistry course, which is basically for focusing on the chemical and the molecular reactions that sustain the life. And we want to understand the chemical functions and the structures, how they work and their mechanisms during the biochemical reactions. And lastly, we want to comprehend the current biochemical literatures based on what we've learned so far here. So here's some important functional groups that we use over and over again. So I want you to revisit all of them and want you to just memorize their structures as well as their properties too, such as alkene, alcohol, ether, amine groups, and so on. And particularly, we're using a lot of aldehyde, ketone, carboxylic acid, or the carboxylate groups, as well as amide or the amino group too. And here are the types of bonds, including the covalent bonds, and most biologically relevant covalent bonds are actually just put together here as the carbon-carbon or the carbon-hydrogen bonding or so, which ranges around here in terms of their energy, as well as relevant non-covalent bonds, which are like a hydrogen bonding or the charge interactions and so on are located here on the bottom of this chart. Now let's get to the thermodynamics part 1. And first we talked about the thermodynamics, what that was for, and that is basically the explanation of how molecules move within the space in terms of the particular conditions that account for the temperatures and the space too. And here were some of the important definitions, the system, surroundings. I'm pretty sure you already know this and are familiar with the terms here. And also different types of systems, such as isolated, closed, or open, which is important to understand better how the energies move into the universe, or not in the universe, but into the system, and exchange between the system and surroundings. And the thermodynamics first law was the energy is conserved and cannot be created or destroyed, but it can be converted from a form to another. And we made an example of the ATP, the chemical energy that changes to mechanical ones as the muscle working and so on. And the energy change can be written as the energy final state minus the energy the initial state. And it also can be written as Q minus W, the heat minus work, too. And here are different conventions that we already covered about. And the heat was related with the endothermic or the exothermic processes, as opposed to work is something the energy changes between the surrounding and the system. And we also talked about the enthalpy, which is the total heat content of a system, can be written as H equals U plus PV, where the PV is the work, the pressure volume work. And the changes of enthalpy can be written as this. What the delta means the changes. And it can be converted a little bit here to get this form that delta H equals QP, where the Q is the heat, for the constant pressure, minus W, the work, plus the P delta V, which is the work too. So it can be also converted to just the QP because of the work minus work here, work minus work. It's going to be canceled out here to leave this QP alone. So that means enthalpy is almost the same as the amount of the heat absorbed in a constant pressure process. And there are different conditions for delta H and what it means. Basically, if it is enthalpically opposed or the favored conditions also. And in the biochemical reactions, in the delta V is 0 because a cell's volume doesn't change that much from the biochemical reactions. So the delta H is going to be delta U. And the second part of the thermodynamics covers the second law of the thermodynamics, which says the spontaneous processes are characterized by convention of order to disorder. And the measure of disorder is the entropy as well as the disorder W2. And we talked about the concepts and how it works actually, which is working in the same trend. And S can be written as Kb ln W, where Kb is Boltzmann's constant and the W is the disorder. And most importantly, the Gibbs free energy explains the spontaneity of the reaction or how the delta S, the entropy change, affects the reaction, direction of the reaction. And the delta G has to be negative for the spontaneous processes. So here's again the very important formula. Delta G Gibbs free energy is the delta H minus T delta S. And here are some conventions and what it means for those functions. And here are the other different combinations of delta H and delta S and how it affects the delta G for the spontaneity of the reaction. And here's the relationship with the delta G, and it has to be negative for the spontaneous or the enthalpically favored and the spontaneous reaction here at all temperatures. And if delta G is positive, of course, it's non-spontaneous and also entropically opposed. And delta G of the reaction can be the sum or all of the delta G of the products minus all of the delta G for the reactants too. And you can refer to the table for delta G anywhere for almost all different kinds of substances and compounds. So you can basically plug in those numbers to get the total delta G for the reaction. And delta G can be written as also delta G naught, delta G at the standard state, plus RT ln of the concentration of all the products and the divided by the concentration of all the reactants. And of course, delta G has to be negative for the spontaneous reaction, and also all other explanations of what the relationship between the concentration of products and the reactants mean in terms of the interpretation of the delta G values and the reaction direction. Here we introduced the equilibrium constant KEQ can be written as this. And the delta G naught is minus ln RT ln KEQ, which is gotten from the formula that we just talked about, and by plugging this 0 for the delta G. And we can move around these variables to get the KEQ equals E minus delta G naught divided by RT, which shows the relationship between the delta G naught and the KEQ. And here's the relationship that shows that just a little changes in delta G naught leads to the huge changes in KEQ values. And here we bring up again the delta G naught equals minus RT ln KEQ. We just leave the ln KEQ to get this formula, which looks like a linear function, similar to the y equals kx plus b, and plugging all these, each of them, into this base type of the linear function, the formula, to get the bent off plot, as you can see here, where the slope is minus delta H naught divided by R, and that also can enable us to get the KEQ values by measuring at the different temperatures, and also can estimate the delta H naught values by looking at these relationships. And then we talked about the standard state, that its meaning is different from the chemistry and the biochemistry, where biochemists modify the standard state a little bit and also use the notation as delta G naught prime instead of just delta G naught. But in biochemistry, we can just drop the prime too. And in that case, biochemist's standard state have the activity almost the same as the concentration because of the low concentration overall for the reactants and products in the cell. And also, the activity of pure water was assigned the value of 1 for simplicity. So you can just ignore the concentration of water anytime for any kind of biochemical reactions that happens within the cell. And also, hydrogen cation activity is 1 at pH 7. As opposed to chemists have this 1 value at pH 0, where the hydrogen cation concentration is 1 molar. But there's no such biochemical reaction happens. It's pretty extreme condition, so we just set it in this way, so it's more relevant, and also make all the calculations simple. And here's again a quick summary within this review session. That here's the indication and how you get the delta U, the energy, the internal energy, as well as the enthalpy changes can be written as this, and the delta H can be plugged in this P delta V, and the work is the same as in the pressure volume work, so you can just cancel out this to get the QP only. And entropy S can be calculated by this, but it's almost impossible to measure the entropy in this way because it's almost impossible to measure the disorder too, which has almost like infinite possibilities. So S can be actually written more, calculated more indirectly by other methods that probably we're going to talk about in next slide. But anyway, free Gibbs energy is a criterion for the spontaneity of the reaction, which can be written as this delta H minus T delta S. And the second summary is about that the predicting spontaneity is different from the predicting the rate or kinetics. And delta G only tells you the spontaneity of the reaction, but not about the rate of the process. And it has to be negative for the spontaneous reaction. At equilibrium, delta G is going to be 0. And delta G can be related to delta G naught using this. And basically the concentration of all the products by concentration of the reactants. So it's the same as this as a general form. And you can plug in the KEQ instead of this products divided by reactants in case of the reaction reaches the equilibrium, and the KEQ, so you can relate the delta G naught to the KEQ in this case. And here's a free energy of a formation that can be added in, and basically add all the delta G for the product minus delta G of all the reactants. So now here's the water part 1. And we discussed about all different types of bonds, including covalent bond, which is pretty strong bonding, as well as non-covalent bonding that comprises ionic interactions, van der Waals interactions, which also has subcategories as hydrogen bonding, dipole-dipole interaction, and London dispersion forces. And particularly this hydrogen bonding is really important for understanding the biochemical reactions, because all the biochemical reactions happen in water as a media, and its strength is relatively high among all these van der Waals interactions. Here we talked about the hydrogen bonding that has a donor group and an acceptor group too, and especially acceptor groups have a lone electron pairs to interact with the hydrogen too. So the donor group has typically hydrogen atom there, which is connected to the electronegative atoms too. And the acceptor is an electronegative atom that has a lone electron pair or pairs. So we talked about the electronegativity here too. And it shares the feature of covalent and non-covalent interactions because it's relatively strong non-covalent interactions. And due to this strong hydrogen bonding, there's a hydrophobic effect that happens when there's a hydrophobes, which are the molecules that do not like the water, or the polar molecules tend to aggregate themselves within water molecule to minimize this entropy increases. So it's also called as an entropic effect rather than just a force. And especially these water molecules make ice-like clathray structures to cage these all hydrophobes. And eventually these hydrophobes will get together to minimize the internal energies, the potential energies there. And here's the structure of the water molecule that has the two lone electron pairs, which allows the interaction, has the electronegativity to allows interaction with the other hydrogen atom within the water molecule for hydrogen bonding, up to 4 of them for each side of these. And here's a quick summary again for the hydrogen bonding. The van der Waals interactions have the 0.424 kilojoule per mole, as opposed to hydrogen bonding have a relatively high energy there, but it's much still weaker than the covalent bonding. And the water molecules is dependent on the orientation of those two water molecules in vicinity, or the neighboring two water molecules. And depending on their orientation, the hydrogen bonding can either weak or strong too. And there's a distance between them that is sort of intermediate between the non-covalent interaction and the covalent bonding. Again, here's an interesting properties about the hydrogen bonding, where the one water molecule can have up to 4 of the hydrogen bonding with other water molecules in vicinity too, by having this sort of the tetrahedral structure. And the ice has an interesting structure that forms the most of the hydrogen bonding that is aligned really nicely between the molecules. But the regular waters can have up to 75% of the hydrogen bonding, so it's more liquidy, I would say, more dynamic and transient in terms of the hydrogen bonding. And also, as another property of water as a solvent, it can dissolve some of the polar and many of the polar substances, such as alcohols, amines, carbonyls, such as ketone or the carboxylic acids, or even aldehydes and things like that. And there are solutions as well as different type of not a solution, but the solvents containing some of the molecules there. So solution is the homogeneous mixture of the pure substances, where solvent dissolve the solute completely. So it disappears, not disappears, but in terms of your visibility, so you're not going to see that actually any molecules that is floating in the solvent when once this solute is totally dissolved into the solution. But the colloid and suspension has the bigger particles that is not dissolved completely into the solution, so you can actually see the light path there when there's a light source nearby. And there are different types of concentrations, percentage, or the molar concentration that you need to understand completely what it means, and you can convert them between the different concentration notations, and also use the scientific notation all the time, which uses 10 to the other powers instead of putting down all the digits. And here are some more important quantities and units that chemists and also biochemists use. Avogadro number, or the 1 mole, is the number of molecules within this unit, and the molar mass, and also the molar concentration, and the scientific notation to simplify the calculations, as well as the atomic mass measurements in daltons and kilodaltons. And here's the definition for the dalton. And here are some more definitions that are important to better understand the properties and behaviors of the molecules. The molecules can have the property as the hydrophilicity or hydrophobicity, and also they can be amphiphilic and also called as antipathic too, which I'm pretty sure you are familiar with at this point. And there's a colligative properties that can happen within the solution, where this property depends on the total number of the solid particles, but not the type of the particles. And it shows the property that lowers the vapor pressure of the solution, decreases freezing points, raises boiling points, and so on. It has the osmotic pressure too, that you can measure it using this simple tool with the semi-permeable membranes and the solution within this column that is surrounded by this membrane. And in living cells, it has to deal with osmotic pressure because there are a lot of stuff, basically a lot of molecules with the higher concentrations within the cell compared to the outside of the cell or the environment. So animal cells become isotonic, meaning that they keep the same osmotic pressure to the environment to avoid any like a burst out of the cells, as opposed to plant cells and bacteria have rigid cell walls, so it doesn't have to worry about osmotic pressure, but it'll hold it really tight, hold the cells really tight there, and avoiding the cells just erupt. And here's a tool. The dialysis is another sort of tool that uses the property of this solution, especially the diffusion of the solutes, where it uses in the dialysis membranes in that the molecules can go through. So it goes throughout to the solvent to lower the concentration within this dialysis membrane. And here's the water part 2. And here we talked much more about the water molecule's properties and its ionization properties and what's its implication on the biochemical reactions too. So water has a slight tendency to ionize to hydrogen cation and hydroxide, but this hydrogen cation is actually hydronium ions, such as this. But the H plus notation is used for simplicity, and we talked about the proton jump, that the water molecule quickly changes to hydronium ions, and that property passes along to the other molecules in vicinity, and that transition is keep going and keep promoting that those changes of other molecules eventually have this proton jump to the other molecules in a long distance pretty quickly. So it's much faster than the direct molecular migration of the proton or the hydrogen cation. And here we talked about the ionization of water in terms of the equilibrium constant, where you can get the equilibrium constant as the ratio of the concentration of the products by the reactants. And there are a lot of just water molecules exist because of only the slight ionization of water. So this concentration of water molecules is pretty constant, so you can put it into this formula by moving this concentration of water to the other side of this expression to get the Kw, which is ionic product. And that is constant as 10 to the minus 14 molar square at standard state. And we talked about the pH and its definition too, and its importance too, and how you can interpret these pH values, make a relation to the concentration of hydrogen cation, and also what's going to be the values for telling the acidity or the being basic of the solutions. And because it's logs, it has huge changes in terms of the hydrogen cation concentrations when the pH changes a little bit. And here are some examples of those pHs. And then we talked about the acid-base equilibria using the Bronsted-Lowry view, where the donating proton or the accepting the proton, the movement of a proton defines that particular substance, if that is acid or base, within the reaction. So here were some couple of examples that I showed you, and interestingly, water molecule can either be base or acid too, depending on the reactions. And for simplicity, water is simply dropped. And acid strength can be measured by the pKa values, which is a acid dissociation constant. It can be calculated by this formula, and this looks a little bit similar to the pH formula too. And the Ka value, which is just the ratio of the concentration of the product by and the concentration of the reactants, not really reactant, but the acid, and its relationship with the pKa is also mentioned during the lecture. And Henderson-Hasselbalch equation is such an important equation that is often used to make a relationship between pKa and pH. And you can basically, starting from this formula, to drive the final one here as written in green, which is pH equals pKa plus log the ratio of the conjugate base and the acid. And when these two values, the concentration of the acid and the conjugate base, are the same, then the log value will become 0. So that's going to be pH equals pKa. And that is actually, we probably were going to talk about it in a few more slides, but when you look at the titration curves, right in the middle point where the inflection happens of that sigmoidal curve, the pH value is the same as pKa, because that's the point where only half of the molecule has been ionized of that particular acid. So here we actually talk about the titration curves. And we talked about the pH and pKa relationship here by the Henderson-Hasselbalch equation. And also the midpoint is where the pH equals pKa, or the concentration of acid is the same as the concentration of the conjugate base. And some other substances can have more than one titration curves. And a good example is in this phosphoric acid, as well as some of the many of the amino acids has 2 or more pKa values. And whenever you see these titration curves, you can actually get each of the pKa values to corresponding ones, and also can calculate the midpoint by where the concentration of that particular acid and the conjugate base are the same. Like I told you briefly in the previous slide, the amino acids have at least 2 pKa's because of these 2 functional groups. And also it can have more pKa values from the side chains too. And Spiteri form is the form of the amino acids, or it can be any kinds of substances where it has the both positive and negative charges within one molecule. Here's the next part, is the nucleic acid part 1. And first, we talked about the nitrogenous bases that gives the identity of those nucleotides, and that is a component or the building blocks for the nucleic acids. And for nitrogenous bases, there are 2 types, the purines and pyrimidines. And this is the structure of the nucleotides, where it has the pentose sugar ring and also the phosphate group and the nitrogenous base that is attached on the opposite side of this pentose sugar, especially the C1 prime and the C5 prime here. And here are the 4 different bases, nitrogenous bases, that you can see in the nucleic acids as adenine, guanine, cytosine, uracil, and thymine. Actually 5 different kinds because of the uracil that is only observed in RNA, as opposed to thymine is used only in DNAs. And you can also should be able to tell the difference between the nucleoside and nucleotide. It's the difference of a presence of a phosphate group that is attached to the pentose ring. And here's the sugar that is pentose, it's 5C. There are 5 carbons as well as the oxygen atom that forms the this pentose ring. And ribose is used for RNA, as opposed to deoxyribose is used for DNA. And the only difference is this C2 prime, where the ribose has the hydroxyl group. The deoxyribose has only the hydrogen atom sticked from there. And you should memorize this structure and the numberings and so on. And here's a difference between nucleoside and nucleotide that we already talked about, the presence of the phosphate or not, or just a hydroxyl group for the nucleoside. And there are different bases that can be attached here using the N-glycosidic bonding. And here's again quick summary for that particular part. There's a difference between the nucleoside and nucleotide. And again, it's really important to remember their names and their structures too, to better understand how the DNA or RNA is formed, and also how those nucleic acids interact with the other nucleic acids, as well as other macromolecules such as proteins. And one important thing to point out is that the nucleotide 5' monophosphate is oxymoron. It really doesn't make sense because nucleotide itself includes the phosphate group, so it has to be said as the nucleoside 5' monophosphate. And the linkage of these monomeric units in the nucleotides can lead to the polymer, the nucleic acid or DNA or even RNA too. And this linkage is done by phosphodiester bond, where you can see the phosphate group, a single phosphate group that is left by having this pyrophosphate or diphosphate group as a leaving group from the NTPs, the building blocks for the nucleic acids. And the direction for reading is always from 5' to 3'. And those nucleic acids, or the string of nucleotides, can form this double helix to make the DNA double helix, where you can see the DNA as a natural form within the cell, as well as in most of the aqueous conditions. And they're antiparallel, so going one strand goes from 5' to 3', then the other, when you look at it from the top, should be the 3', and then the bottom should be the 5' too. And it's right-handed under normal condition. Here's this cartoon that shows the interactions between the nitrogenous bases within the nucleotides, and the GC have the 3 hydrogen bonding that are formed between them, as opposed to AT has only 2 hydrogen bondings. So GC has a higher energy that is kept there, so it's hard to tear this apart compared to the AT interactions. And here we're going to talk about some biochemical tools that are important for analyzing the nucleic acids. The first one was a gel electrophoresis that I explained in last lecture, that you can place the gel, either agarose or the polyacrylamide gel, and then put down the samples there, and using the electricity, you can see the migration of those DNA fragments, and their migration speed, or the rate, is dependent on the size of DNA fragments or the RNA fragments. And the molecular cloning is another very useful tool that is used every day in the labs. And it uses the restriction enzymes as well as ligases to prepare the two different DNA fragments, or either the vector or the plasmid. And then you cut the regions that you're interested in and insert it to the cloning vector to make the chimeric DNA, and that will be later inserted into the bacterial system or some other cloning system to amplify these DNA fragments. And another really important biochemical tool is polymerase chain reaction, or PCR, where it uses in the polymerase and the primers, two primers, that defines the starting and the end point for the amplification of that particular gene. And by using the multiple cycles, it amplifies basically the DNA fragments over and over again to make bulk of them, or either analyzing them or using them as a material for another molecular cloning, and so on. And using the chain terminal sequencing, or the Sanger sequencing, you can tell each of the nucleotides that are formed, or that consists of the particular DNA fragments that you have. And it uses the dideoxynucleotide, which cannot form the polymer anymore. So that's why it's called a chain terminator. So it's not going to be linked. There's no place to get the phosphodiester bonding anymore there. So you can have sort of a random stoppage of all these fragments with the different size. And since you have all the different type of labelings, like a fluorescence light, depending on the different dideoxynucleotides, you can actually tell that the size and also the particular nucleotide that is attached at the end, at the 3' end of the fragments. So you can read it from the shortest one to longest one, basically, to identify the sequence. And TM is an important property of the double-stranded DNA to regulate some biochemical reactions. So TM, the melting temperature, can be controlled or modified by the GC contents, or the concentration of the salt, the length of the sequence itself as well as the gaps, if they're present, between those two strands of the DNAs. And here's the second part of the nucleic acids. First, we talked about the primary structure of DNA, which is basically just a linear sequence of the bases from 5' to 3'. And the secondary structure of DNA too, which shows the double helical structure of the DNA. And there's hydrogen bonding between the bases, and also van der Waals interactions contribute to the stability of this double helical structure by having this base stacking forces too, especially from G to C and so on. And there are major and minor groups in double-stranded DNA, and it has important roles, especially for the major ones, and that is exposed into the solvent, which is water. So the soluble proteins and some other compounds can be physically interacting with the base pairs or the nucleotides within this major group. And we talked about the different types of DNA double helices. There are three major forms. The bDNA is the sort of widely observed form that can be seen under a normal condition within the cell, and it follows the Watson and Crick model, as opposed to A form is shorter and broader, but it happens more often in the double-stranded RNA or the RNA-DNA hybrids. And the Z form is opposite-handedness, and it happens only under special circumstances. And these 6 torsion angles, starting from alpha to zeta, describes the conformation of the nucleotide within the nucleotide chains. And also this chi angle explains the orientation of the base too. And also there are syn and anti in terms of the orientation of the nitrogenous bases attached to the sugar, or pentose sugar. And the pentose sugar also have a different conformation by having the C2' or C3' puckering, which are called as C2' endo or C3' endo respectively. And they can be seen in different types of DNA structures. And there's a superhelicity that you can see within the DNA, especially the DNA is circular, and it's also called the supercoiling that makes this kind of supercoiled structure of these circular DNAs. And it's caused by the overwinding or the underwinding of the DNA, and can be released by nicking one of the strands of that double helical structure of the DNA. And the most close to circular DNA is supercoiled, looking like this. And topoisomerases are basically the enzymes that change that number of supercoiling of the topoisomers of the DNA. So it basically changes the superhelicity. And there are 2 different types of the topoisomerases, topoisomerase 1 and topoisomerase 2. And they use different mechanisms to achieve the inner conversion of the topoisomers. And we talked about the hierarchical organization of the chromosomes, starting from just DNA double helix to the nucleosome, where these double helix are wrapped around the histone proteins, then 30 nanometer fibers, and also the more higher order structures too. And the packing ratio was changed from 6 in nucleosome, 40 in 30 nanometer fiber, to the 10,000 in the mitotic chromosomes, where there are the maximum packing happens. And in addition to DNA, we talked about the RNAs too have a slightly different bases. A, G, C are the same, but the uracil is the difference there because the DNA has the thymine instead of uracil, and there's some properties related to that, and as well as the sugar type of the sugar is different between the DNA and RNA too. And here are some other types of RNAs, such as transfer RNAs or the ribosomal RNAs, where the secondary structure has an important role in terms of their stability as well as the functionality too. So here's the example of the secondary structure where you can see the loops and the hairpins and the helices and so on. And also the tertiary structure too, which is also looking different than this just secondary structure, where it has some interactions like hydrogen bonding from the one helix to the other helix or the loops too. And those interaction is really, again, important for forming the tertiary structure of the RNA, which can have a particular functions and critical functions for the biochemical reactions. And that kind of leads to the idea of RNA world hypothesis, because RNA has all the functions, not only having the genetic information, just like DNA, but also have a catalytic function too. So it catalyzes the biochemical reactions a lot, like the enzyme proteins too. So might have been just the RNA was the initial form of the life, but succeedingly, the descendants have the integrated the DNA and proteins into their mechanisms to more sophisticate their system. So here's the last part for our review session, which is talking about the protein part 1. And we discussed a lot about the amino acids, the type of amino acids and how they form the polypeptides, as well as their properties and so on. And you should, again, remember all different types of amino acids as well as their functions and their critical functional groups, such as like the hydroxyl group on the side chains and so on, and how those amino acids interact with other molecules and their properties, such as like polar or non-polar, acidic or basic properties and so on. Anyway, amino acids, especially in biochemistry, refers to alpha amino acids, where it has the alpha amino acids connects the amine and the carboxylate group, and there's a hydrogen atom that is attached, as well as the R group, which is a functional group. And especially by interacting this amine group or the amino group of the one amino acid to the carboxylate group of the other amino acid, it can form this peptide bond to make the polypeptide. And when you have the long array of this amino acids that are all connected and formed into the globular shapes or the certain like functional shapes, then it will become a protein. Like we discussed earlier, the amino acid has at least the 2 pKa value, which is pKa1 and pKa2. Or you can also just drop the A and call it as pK1 and pK2. And they have different values here. One's more acidic, one's more basic. And also, there could be other pK values from the side chains too. That'll make this titration curve even more with more steps. And here are the expressions that you can get PI, which is isoelectric points. And this PI is basically the average value of the 2 pK values within this is virion. And for the basic amino acids such as lysine, arginine, and histidine that has the basic group, the PI can be calculated as keeping the pK2 here, which is more basic one, but use the pK for the side chain, which is relatively basic than the pK1 value, and get the average of that. And for acidic ones, it's the other way, that use the pK1 and pK from the side chain, which is relatively more acidic than the pK2, to get the pI value by simply getting an average of those two pK values. And protein purification uses this pK or the pI values because it has minimal solubility when the pH is equals the pI, so it'll have the most precipitation of that particular protein. So you can use this pH when you know the pI for that protein of interest to maximize your yield of that protein purification. And also there's other technical, the isoelectric focusing also uses the pI value for separation of the proteins. And here are the 20 amino acids that we talked about in terms of their properties and types. There are 9 nonpolar, 6 polar uncharged, 3 basic, and 2 acidic amino acids. And you must memorize all of them, the structures, the single letter, 3-letter codes, and so on, their side chains, and so on. And here are some examples here. I'm not going to go over all the amino acids and their properties, but there are other grouping here. One is aromatic that has this tryptophan, phenylalanine, and the tyrosine have this aromatic rings, the phenyl rings. And also sulfur-containing amino acids are the cysteine and methionine. And cysteine is particularly interesting because it can form the disulfide bridges with other cysteine in the other polypeptide. And here are the how you memorize the one-letter code for amino acids. And here's the first summary. The hydrophobic amino acids are insoluble in water and tend to cluster on the inside of the protein because of the hydrophobic effect. And those amino acids include the nonpolar hydrocarbon containing on their side chains containing amino acids, as well as the amino acids with the aromatic side chains. The second summary for that is that hydrophilic amino acids have ionized or polar side chain groups that can participate in hydrogen bonding, and this hydrogen bonding is achieved by the other polar molecules, or even just a water molecule too. And these are the examples such as acidic or the basic amino acids. And they're usually bound on the surface of soluble proteins because they can readily interact with the water molecules. But it can be also buried inside of the protein or the one side of the protein too. But that is mostly for the interacting with the other polar molecules, such as protein or other residues inside of that protein. Enantiomers is basically the molecules that have chirality, and that's a mirror image of those two initially identical molecule, but have a different base for them. And there could be a racemic, which has a 50-50 of those enantiomers. And in that case, it will have a property of the plane polarized light of 0, because half of them will try to turn it one way, but half of the other enantiomers will try to turn it on the other way, so it'll cancel out to have a 0 angle change of that plane polarized light. But there's only L enantiomers of amino acids are made in the cell, and that is relevant to the activity and the stereospecificity of the enzymes. And here we talked about the Fischer conventions and Fischer projections that you need to understand too. And we talked about the Korn rule to tell that if the that compound is in D or L isomeric form. And basically you can use this Korn rule to place the hydrogen atom away from you and see that if the Korn is red in the clockwise here, or counterclockwise. If it is clockwise, it's the D form, and if it's counterclockwise, that's the L form. And here it shows the RNA codon table, which is used to interpret this messenger RNA and how it is going to be translated to the protein or the amino acids through the transfer RNA. And we also talked about the peptide bond formation, where the carboxylic acid group or the carboxylate group and the amine or the amino group are interacting with each other and pop out the water molecules there to form the peptide bond. And there are some modifications on the side chains too that are important for the some biological functions, biochemical reactions too. So examples were so it's called as a post-translational modification too. And also we talked about some examples such as hydroxylation of the proline. And also sticking in some of the hydrophobic regions within amino acids leads to the changes in localization, such as the hydrocarbons interacting with the other non-polar substances within the cell, which is phospholipid membranes and other substances too. So it can be stuck in there. And finally, we talked about the non-protein amino acids, which have biologically important roles, and several of them are the neurotransmitters. And we also talked about one example of how this non-protein amino acid is synthesized too. So this is it for the review session. And if you still have any questions or if there's anything that is unclear, just email me or come to the office hour. So good luck with the midterm exam 1.